So the fifth lesson in your ecology unit will have been looking at one of the required practicals, which is about field investigations, which simply is how you go about sampling and estimating the number of a specific organism that is in an ecosystem, or the spread of it, the distribution of it, where it actually falls and lives and exists within an ecosystem. Now, the key tool you will have used is a quadrat. Now, quadrat is simply a 1 meter squared metal frame that is split into smaller squares itself, usually. Now, what we will have done within the lesson, what you will need to make sure you are confident with, is the methodology of how to use a quadrat to estimate the total number of a specific organism within an ecosystem. So the first thing is that this quadrat will get placed randomly within an ecosystem, and the simplest way in which to do that is through splitting up the area in which it's being used to sample into a grid and generating coordinates randomly, maybe using a random number generator, that you can then place that quadrat at across the entire ecosystem. Once that has been done for a specific number of times, let's say 10 or 20 times, each time that's done, the quadrat's placed down, and then the number of the organism that's wanted to be sampled, so for example, if you look at the picture, buttercups or daisies are counted, and those results are recorded for each coordinate for the number of that species. So let's say the number of daisies here. Once you've done that for the 10 or 20 times you place the quadrat down at the coordinates that you've generated, you can then work out the average number of organisms per quadrat, per 1 meter squared. So you count up all your number of daisies and then divide it by 10 or divide it by 20, divide it by how many times you've used and placed the quadrat down. Once you've got that average number of organisms that you're trying to sample per quadrat, the way in which you then take that number and use it to estimate the number of organisms within an ecosystem is you simply take that number and you multiply it by the total area in meters squared of the ecosystem, which will give you an estimated number of organisms that are contained within it. Now, this methodology is not perfect, so it is only an estimate. It's a scientific guess, as it were, for how many of that organism are contained within that ecosystem. There are different issues with this as a methodology, such as it wouldn't really work particularly well if you were trying to sample the number of a living, moving organism, such as an animal, because obviously they just walk out the quadrat. But it's particularly good for sampling plant species or species of organisms that stay still. You will also use quadrats to sample the distribution or the spread of an organism across a particular ecosystem, and that is using something called a transect line that your teachers will talk more in depth with you about, because it's quite difficult to demonstrate on a video. Otherwise, that is your key steps for carrying out field investigations for the required practical that you need to know in terms of how to use a quadrat.